I am Dr.Ashok Kumar, Head Department of Zoology and Industrial Fish and Fisheries, Ganga Singh College, Chapra, running under JP University, Chapra, Bihar. I am going to explain, as I explained in my last class, the difference between the post-mortem and rigor mortis. Rigor mortis that indicates the freshness of the fish muscle, and this rigor mortis is sustained contraction of the muscles, transverse muscles, and it continues from. It may be enhanced by certain methods, and during rigor mortis, the stiffening of muscle takes place, and this stiffening of muscle shows that the fish flesh is under the fish flesh is the very fresh, and now how factors which are responsible for prolongation of the duration of rigor mortis. Then first is how duration of rigor mortis, duration, how to increase duration of rigor mortis. That is, the first thing is rigor mortis differs the autolytic changes. The autolytic changes, spoilage and bacterial spoilage of fish, and the following steps are taken for enhancing the period of rigor mortis. That is, first is lowering of the temperature. Lowering of the temperature is done by keeping the fish in ice, or in freezing, or by freezing, or keeping the fish in ice. That is also called glazing. So lowering of temperature is done, and hence there is this one is important method, lowering of temperature, either keeping in the ice, or keeping in freezing chamber. Second is mode of packing. That is, the slaughtered fish delays the rigor mortis. The slaughtered fish. Third is mode of fishing. Fishing operation is done with minimum stress, minimum dragging of the fish, and handling during the period of rigor mortis. During the period of rigor mortis, less handling, that keeps the fresh. Further, the dragging during dragging or handling, the the rigor mortis slows down, and after rigor mortis, spoilage of fish flesh takes place. So this is more. So the minimum, the use use of anesthesia or electric shock in the net, or on the board of fishing vessel. These are the two things. First is electric shock in the net, or after catch on the board of the vessels, and second is anesthesia. Anesthesia, that is the injection given to the fishes, and the anesthetic compounds are sodium amytal, chloral hydrate, Novocaine, and amobarbital, sodium thiouracil, urethane, 2-phenoxyethanol, and quinaldine. These are the anesthetic which are given in the form of injection, and this also increases prolong the rigor mortis. Now after, so mode of fishing is also important for prolongation of the rigor mortis. Now one thing I would like to explain, that is the post rigor spoilage. After, as the rigor mortis finishes, this, as I had already explained in my previous classes, that rigor mortis ranges from 30 to 120 hours. After 120 hours, if after 120 hours there appear the fish flesh undergoes rapid or intense chemical alteration, chemical changes, and autolysis becomes inedible with foul smell, and the flesh becomes inedible with foul smell caused by chemical changes or autolysis. That is, that chemical changes and microbial attacks, means attack of bacteria. Chemical spoilage occurs due to rancidity. That is, rancidity means unpleasant smell and unpleasant taste. This is rancidity. Unpleasant smell and unpleasant taste, and this occurs due to the decomposition of the chemical compound of the flesh. So rancidity, it is oxidative rancidity. Oxidative rancidity means that it is due to the rapid oxidation of lipids. This is rapid oxidation of lipids in katla, rohu, mrigal, and fatty fishes, etc. How this oxidation takes place? The antioxidant in the live fishes, that is tocopherol, that is a derivative of vitamin E, tocopherol. That antioxidant is tocopherol, and this tocopherol now this after death, the supply of antioxidant, that is tocopherol, that stops. Hence oxidation takes place. Further, the heme is present in the hemoglobin, and thus that when there is no antioxidant in the form of tocopherol, then oxidation of fatty acids takes place, and that is enhanced by hemoglobin of hemoglobin, that becomes ferric or ferrous, that helps in oxidation of the. This results in oxidative rancidity, which changes the color of the flesh from light yellow to rusty iron, that is brown, the rusty iron, and the lipid or the oil which remains thin after oxidation. The lipid or fatty is the lipid or oil after oxidation becomes thick and viscous. So after oxidation, after oxidation of the fat or lipid, as we know that the PUFA is the major PUFA, polyunsaturated fatty acid, that is which is oxidized and that remains present in the fishes. This PUFA, polyunsaturated fatty acid, and that fatty acid, that fatty acid becomes oxidized, and the color of the fat lipid changes from light yellow to rusty iron, and the lipid which was very thin now it becomes thick as well as viscous after oxidation. The storage of fat with highly unsaturated fatty acid, it is the fat with highly unsaturated fat is CUFA is present, polyunsaturated fatty acid is present, and hence that fatty acid that is the real problem of fatty acid oxidation is the real problem which the fishing industry has been facing from the last century. From the since its inception, from the very ancient days, this problem has been is in process. Other factors that is responsible for. So first factor is oxidative rancidity. It is due to the oxidation of fatty acid, and other factors which are responsible, that is the presence of aluminum, calcium, magnesium, zinc in salt used in salt curing. In salt curing we keep the salt in one layer, then next upper layer that remains fish, and that fish remains degated and no organ eviscerated, and that we place one by one in on the just above the ice layer. Again we put ice layer, again we put now fish flesh there, again fish. So in this way it is salt curing is done, and during fish salt curing during salt curing we keep the salt. That salt contains magnesium, calcium, aluminum and zinc, and that causes the rancidity, the onset of rancidity, that salt with MgAlZn and Ca, that is responsible for. And further, copper and iron or their salts, they also cause oxidation of fatty acid, as copper and iron acts as catalyst in fat oxidation. So in any processing plant, wherever you go, you will get enamel, that is not where the fish is kept, that is not in the pot of made up of iron or steel or copper or anything, and such type of. Further, copper and iron, they also act as catalysts, so they are also responsible for oxidation of fatty acid. Precaution, how to control rancidity, how this oxidative rancidity, oxidation of fat, how the precaution we take. So first thing is we keep the fish in polyphenols, in that aqueous solution of the polyphenols, where we keep, or glazing of fish with ice containing antioxidants. The ice containing antioxidants, either keeping in antioxidant like aqueous solution of polyphenol, or if this polyphenol or antioxidant is kept in the ice, is kept in the ice, in ice it remains there. Further, the second is the storage chamber of the fish on the vessel, that must contain nitrogen or carbon dioxide. Third is pure salt, that is pure salt, that is sodium and chloride. Only sodium and chloride should be there in that salt, not along with the magnesium, calcium, Zn, iron, these things. That should not be there. So only pure salt without any impurities should be used for salt curing. Next is the fish should be subjected to deep freezing. Deep freezing means minus 20 degrees Celsius, the deep freezing. Now, autolysis. Autolysis is the accumulation due to the accumulation of lactic acid. Due to accumulation of lactic acid in the muscles, it becomes the lactic acid breaks the wall of the lysosomes and lysosome, the membrane of the lysosome breaks due to the acidity of lactic acid, and these are high the lysozymes, they come in the muscle which was lodged in the inside the membrane of the lysosome. Now these are hydrolases, these are peptidases, proteinases, lipases. These are the enzymes. They come out from the lysosomes, and they come out from the lysosomes and they act on their substrates, and the step-by-step breakdown products of proteins are the amino acids. Step-by-step breakdown products are amino acids, guanine, hypoxanthine, etc., and their further decomposition into, now protein was broken into amino acid, guanine and hypoxanthine. Then now this amino acid is converted into ammonia, NH3, ammonia, CO2 or volatile basic compounds. These are amines and fatty acids, and these amines are again smelling products, and basic compound amines and fatty acids, and foul smelling products like indole, escatol and H2S. Indole, escatol and hydrogen sulfide gas that smells like rotten eggs, and hence it is this render the fish unpalatable. Histamine, that is amine, as I had already explained, that protein converts into amino acids by protease, and now it converts into ammonia, it also converts into amines, and histamines, these are found in blood. These histamines are imparts a bitter or a pungent taste with advanced spoilage. Now, as the spoilage advances, the histamine produces a very pungent smell or bitter and pungent taste and bitter taste with advanced spoilage. The bitter tasting hypoxanthine is also produced as hydrolysis product of ATP. So hypoxanthine is produced as a hydrolysis product of ATP, adenosine triphosphate, and hence ATP converts into ADP, ADP converts into AMP, AMP converts into IMP. IMP means that is inosine monophosphate, inosinic acid or inosine. Now IMP plus hypoxanthine. This hypoxanthine provides bitter taste. Further, microbial spoilage also takes place. Now microbial spoilage takes place, and this microbial macromolecules. Now the macromolecule. In next class, I will explain about the microbial spoilage, and this is very important because here anaerobic oxidative oxidation will oxidative processes will occur, and hence it results into acids, volatile and non-volatile compounds. Spoilage is nothing. It is simply the undigested. Simply, it is actually a spoilage is in essence nothing more than partial digestion of food by bacteria. Partial digestion of food by bacteria, and this is called this is called spoilage. Partial digestion of feed of bacteria. So food by bacteria, that is spoilage. Thank you. In next class I will explain next again go into the microbial spoilage in detail. Thank you.